Okay, so chapter 18, now we're starting, we've broken down fats, we've broken down sugars, now we're breaking down proteins, essentially amino acids, because we're assuming that the proteases are broken it down. So now we're going to actually take apart the amino acids and scavenge from that. So this is the very last biomolecule that we have to aid in the energy production. I like to park some recreation, even though I don't eat a lot of meat, but I thought it was apropos for here, for high protein and all the amino acids. Okay, so where did it go? All right, so one of the main parts of the amino acid that we have in order to be scavenged and reused, okay, so there are only three parts to an amino acid, right? We have what makes an amino acid an amino acid. Oh, whoops. Ooh. We have a what? We have an amino group. We have a carboxyl group. And then whatever, yes, because hydrogen itself isn't useful. So we've got these three parts that we can use and utilize for energy or for parts or what have you. Okay. First off, the amino acid has to lose the amino group. Okay. So we've actually seen this before because we're going to form what's called an alpha keto acid. And we've talked about 3 alpha keto acids so far. What was one of the alpha keto acid amino acid partners that we've done? Alanine is a partner too. What? Because alanine is an amino acid and it makes alpha keto acid. Pyruvate. Okay. And so then we also had aspartate. You can remember what was aspartate's alpha keto acid? No, aspartate's not glutarate. It's really important. It's in multiple metabolic pathways. Oxaloacetate, OAA. And then we have glutamate, and glutamate's alpha keto acid is alpha ketoglutarate. Okay, and so those are the 3 that we've known so far. Okay. But before we do pretty much anything, you've got to get rid of that amino group. Then we have different fates for the carbon skeleton. One is you can try to use up everything you can to ultimately make the carbons down to carbon dioxide. So like what we did with the fats, when we kept breaking them down, until it gets to acetyl-CoA. Acetyl-CoA then ultimately can go on to make carbon dioxide. We can also provide 3 to 4 carbon units, like chop them up into little pieces that can be converted via, we'll talk about the pathway, for glucose for the brain. So you can actually kind of chop it up into smaller pieces to make glucose. Or you can also have them enter into citric acid cycle, okay, which we kind of already hinted at. Like, for example, aspartate can enter into the citric acid cycle as what alpha keto acid again? Aspartate? Oxaloacetate. Okay, so, and glutamate makes alpha ketoglutarate. And then alanine can make pyruvate, which is technically would be like step 0, right? And so... Okay, so this is just the broad strokes. Okay. And where we're going with all of this. Okay, so we have our proteins. What class of enzymes, they're not written here, but what class of enzymes will break down the proteins down to amino acids? Proteases. Then, and another thing is you can also get those from dietary proteins, and proteases can break them down. What we have to do is we separate the amino group. And then depending upon the species and stuff like that, there are ways to get rid of the amino group. Or you can use the nitrogen to build other things, which we'll talk about that later on as well. And then we take those carbon skeletons to make alpha keto acids. Alpha keto acids could enter directly into the citric acid cycle. Or, with respect to aspartate and also, I guess, alanine, you can go directly into gluconeogenesis too, right? Because we have OAA or pyruvate. We also have something that this is a new one that we haven't heard of before, called carbamoyl phosphate, which is used with nitrogen, which enters into the urea cycle. Okay, for those who have urea as the excretion. Not all species have urea, which I believe mine's off of some students in the past when we talked about how the fact that some species don't urinate, they don't pee per se. I mean, there are some people that just like how many times have you seen a bird hike a leg? You know, because they don't. What do birds do? They don't make urea. Does anyone know? They're uricotelic, which means they make uric acid. And then fish, many fish, not all fish, but many fish just excrete ammonia. And so they don't have to do this entire thing. But we're going to find out that it's not nearly as pretty as what this looks like. It's called the aspartate argininosuccinate shunt of the citric acid cycle, which is a mouthful. But once you see it, it's like, oh, okay, now it makes sense. Hopefully, that once you see it, you'll say, now it makes sense. All right. I really like. So I really like this figure from early on. Once again, we have proteases. This is just showing you, here we're talking about liver cells, hepatocytes. So we have our amino acids. They got broken down we got our amino acids from proteases. They come in. We convert all amino acids to their alpha keto acids. This is very important. Glutamate is a major player, glutamate and alanine. We're going to talk about how that plays, okay, here. But glutamate is special because I'll just tell you right now, every single, what is this enzyme right here that's moving an amino group here to make glutamate and alpha keto acid? It's aminotransferase. Every single aminotransferase utilizes alpha ketoglutarate and glutamate. So that means glutamate cannot use an aminotransferase to get rid of its nitrogen because it's always made by all of the other aminotransferases. Okay, but we make the alpha, but there still is a way to get rid of it. We make the alpha keto acids, and then so on and so forth. We already talked about how these can be broken down, go to citric acid cycle, gluconeogenesis, and so forth. The nitrogen gets kicked off, and we'll talk about how that happens in a moment. We have glutamine, which is a major player, and we'll talk about why not today, maybe on Friday. But then the nitrogen here is either going to be, for some species, just emitted as ammonia, for some like us, for urea, and then some is uric acid. And that's where, like I said, I kind of already gave this away, but the ones that do ammonia is called the monotelic animals. So we're talking about mainly a lot of the fishes that do this. Some of the larvae will do that. There's ureatelic animals, which are, humans and mammals and things like that. But sharks urinate. And so that's a little factoid for Jeopardy, I guess, you can one day use. But perhaps. But yeah, so sharks undergo urea, but we also do urea. And then birds and reptiles they make uric acid, so they're called uricotelic animals.
Do we need to know the structures of it?
No. You may, ammonia. And for urea, by the end, you may know it just because of after seeing it and hammering it over and over and over again. I would never ask you this one. But one thing I want to point out is, with the exception of ammonia, if you look at both urea and uric acid, all of the carbons are highly, highly oxidized. Why is that important? For urea, it's completely oxidized. It can't get any more oxidized. Why is that important?
We have all the energy.
We got every bit of energy from that carbon we possibly could get, so now we're going to get rid of it. Okay, so we're not wasting. And so that's why it's important. Just like the fact that that's why we take those saturated fats or even unsaturated fats and ultimately make it down to carbon dioxide. Okay. Actually, really kind of already given away the answers on this one. So the class of enzymes that degrade proteins, once again, were called proteases. Yes. And then we have the amino acids that lose their amino groups by another type of enzymes. With glutamate is one exception. So besides glutamate, all the other amino acids are losing their amino groups of their backbone, at least, by what class of enzymes? They're moved using aminotransferases. And the aminotransferases are named after the amino acid other than glutamate. So, for example, alanine aminotransferase, which is one of your liver biomarkers, is the one that utilizes alanine to pyruvate. And it's because it's a ping pong mechanism, which we'll see in a little bit. And the other partner is alpha-ketoglutarate into glutamate. Whereas serine aminotransferase would be serine, and it's alpha-keto acid. And then, of course, it also does glutamate and alpha-ketoglutarate. All right, so we have our aminotransferases. What they do is they're going to remove the amino group, and it always results in an alpha-keto acid, and it's very important that it's reversible. These are reversible. Okay, so they go frontwards and backwards. That means that their KEQ is going to be approximately 1. It just means, the fact that it's reversible. And I wouldn't ask you this now because we barely glossed over it, but that means that delta G prime is going to be about 0. It's really low, right? Because it's right roughly at equilibrium. All of the amino acids will transfer their amino group to alpha-ketoglutarate. And so, just as a review, once again, what amino acid is formed from the alpha-ketoglutarate? Glutamate. And when you're going backwards, you go from glutamate to alpha-ketoglutarate, and you'll make whatever the other amino acid is. Every single one has the exact same mechanism. They all use the same prosthetic group. That prosthetic group is called PLP, which stands for pyridoxal phosphate. And I didn't check in the most recent edition of the book. I don't know if it's actually 18.6. It's really obvious what mechanism it is. Because what we'll find out is that it actually makes 3 different types of reactions. Okay, I want to get that far, and it's probably where I'm going to leave off for the day, but this is what PLP or pyridoxal phosphate looks like. Normally it's an aldehyde. So normally it has an aldehyde, but it can also become an amine. And so that's how the nitrogen gets moved. Okay, and we'll talk about the reason. But normally it's an aldehyde. And the mechanism isn't that hard. It's just the fact, especially if you write it out a couple times and you understand what's going on, especially for the aminotransferase, it's just the fact that there are multiple steps to it. Because you do it's 4 steps, and then you have to do the exact reverse of the 4 steps to go backwards, right? So, it literally is reversible. So, okay. So what happens here is that the enzyme, the aminotransferase, has a lysine, which is important. Of course, lysines are great nucleophiles. And they're not showing, but if you remember from organic chemistry, that can come in and smack that carbon, and it breaks that bond. The first time, it would make an OH. But since it can actually attack it up to twice, when it attacks it that second time, it's going to pop it off as a water. And so now we have what's called the Schiff base. We have that imine. This is an imine. So the pyridoxal phosphate, the aldehyde, has now become an imine that's covalently attached to the lysine of the enzyme. And they just call it that Schiff base because it's named after Schiff. Because we see these over and over and over again. And these Schiff bases can do multiple types of reactions. This imine can. So, whoops. So what we have to do, or this is where I'll do this one, and I think the next slide. Just because the next slide is just a lot, right? But all you'll see in a moment. So these are the 3 types of reactions. This is an important part. They can do the aminotransferase. Okay, that should make sense. This one, if you think about it, is easy too. It does what's called racemization. Because remember, The amino acids, we talk about typically L amino acids, but there are some that you can have that are D, and in particular proline, since you have to break it and open it and break it and open, to get to move. And so you can go from one stereoisomer to the other. So it's called racemization. So all it does is it attacks the same amino acid, and then it just goes backwards, but it comes from the other side. So it makes a D to an L or an L to a D. And then finally, the one that's the trickier of the three to remember is the decarboxylation. We can actually get rid of a CO2 group. Okay, all of these, you form a shift base and what's called a quinoid intermediate, which we're going to see some of these quinoids and things like quinone and things like that in a couple of chapters. This isn't the only one. But these are the three types of reactions that are used. One is the aminotransferase, racemization, which is just a fancy word of switching between stereoisomers. And then finally, we can do decarboxylation, where you can actually get rid of that carboxylate group. And this is the figure, like I said, it used to be figure 6, but I know it's still in there. I just didn't write down. And it's not the easiest one to follow. These are the steps. This is for the aminotransferase. This is to do racemization. And then this is the decarboxylation. And I have them broken down on future slides, so that way you don't have to try to look at it really tiny print. But this is just to get an idea of where we're going to go on Friday's class.